Welcome back to the Health Longevity Secrets Show with your host, Dr. Robert Lufkin. His new just-released book, titled Lies I Taught in Medical School, has been an Amazon bestseller for over 3 months in a row and the #2 best-selling book on longevity. Here is a short video of a recent book launch celebration from Kalika Yap.
She was just a girl.
We're like diamonds in the sky.
Don't you worry, child.
Yeah, except for me. Starships were meant to fly.
'Cause it's always a good time.
If you're enjoying this program, please like, subscribe, or leave a review. It will help others to learn about our work. And now, please enjoy this week's episode where we look at rapamycin for longevity with Dr. Matt Kaeberlein.
Hi, I'm Dr. Robert Lufkin. Imagine unlocking the secrets to a longer, healthier life. Well, that's exactly what we're going to do this week with Dr. Matt Kaeberlein, a pioneer in longevity medicine from the University of Washington School of Medicine. This episode promises a journey through the realms of aging biology, where we unpack the aspirations behind Dr. Kaeberlein's dedication to this field, tracing back to a pivotal seminar with MIT professor Lenny Garanti. As we age, the pursuit of extending our health span becomes deeply personal, and here we contemplate the real-world applications of this research that could define our golden years. From Easter Island soil to the forefront of anti-aging science, the drug rapamycin emerges as a beacon of health. Our conversation with Dr. Kaeberlein sheds light on this fascinating compound and its ability to possibly replicate the life-prolonging effects of caloric restriction, but without the dietary sacrifices. We navigate the complexities of aging interventions, from lifestyle tweaks such as diet and exercise to the nuances of cutting-edge pharmacological breakthroughs. This episode is a testament to the intricate dance between the choices we make and the science that could help us live not just longer, but better. I'm proud to be hosting this summit as 25 world-class anti-aging experts reveal new science and breakthroughs you can use today to reverse the aging process in every aspect of your life. The free virtual summit starts June 20th, 2024. Please click on the link in the show notes to reserve your spot. This episode is sponsored by Cyfox Health. One of the most common questions I get asked is which blood tests I check myself on a regular basis. Well, I check 17 biomarkers, but the secret is I'm able to do everything from the convenience of my home. I collect a sample with just a simple finger stick like this. You don't need to be a doctor to do it. Then I just mail it in and wait for the results. Cyfox Health makes it possible. Use the link below to get 15% off. This episode is brought to you by L-Nutra, maker of the Prolong fasting mimicking diet. If you'd like to try it, use the link in the show notes for 20% off. And now, please enjoy this week's episode. So delighted to have Dr. Matt Kaeberlein, who's a professor at the University of Washington School of Medicine. Matt, thanks for joining us today.
Sure, thank you. It's a pleasure to be here.
Before we dive in, maybe you could tell us a little bit about your background and how you came to be interested in this fascinating space.
Yeah, so I actually got interested in the biology of aging at the beginning of my graduate career. So I went to MIT where I did my PhD, and during my first year at MIT, I heard a seminar by a professor there named Lenny Guarenti, and he was talking about how his lab had just within the last couple of years started studying the biological mechanisms of aging. And I don't, to this day, I don't really know why that resonated with me, but I was so fascinated by the idea that you could use genetics and molecular biology and biochemistry to study something as complicated as aging that that I went and I talked to Lenny and ended up joining his lab for my PhD thesis, and really haven't looked back. And so, you know, the specifics of what I've been studying in my own work have evolved, I would say, over the, you know, the 20 plus years since I heard that seminar, but it's all been related to the biology of aging. And you know, I would say the one thing that has changed is back then I was young enough that I didn't really appreciate the sort of personal importance of this. But I would say that as I've gone through my my career and gotten a little bit older, I have also transitioned my interest into, you know, maybe a little bit more of the immediacy of what can we do about the biology of aging in people to improve health span, hopefully lifespan, but really, you know, keep people healthy as long as possible by modifying the aging process in a positive way.
Yeah, that's a great observation. How many people in this space that are in, you know, medicine or healthcare, that as they get older and older, they sort of transition their interest to longevity, and there's an inordinate focus on it out of self-interest.
Becomes a little more personal, absolutely.
Well, the lab you worked in at MIT, Lenny Guarenti's lab, has a amazing reputation of a history of great workers in that, great people in the lab who've gone on to have amazing careers on themselves, including yourself, Dave Sinclair, Brian Kennedy, Gil Blander. I wonder, what do you think? What was it about that lab? Was it Lenny? Was it you guys were just especially smart guys, or was there the timing was right, or what was the magic sauce, do you think?
Well, I certainly would not claim that it's because I'm particularly, you know, a genius or anything. I think that, so what I would say is I think it's a combination of factors, right? I think Lenny absolutely ran the lab and was a visionary in lots of ways, but ran the lab in a way that allowed people who were very self-motivated and risk takers to flourish, right? I think that was part of it. I think it was also, you know, when I was in the lab, it was really an amazing time because Brian had just left, but I knew Brian, he was still in Boston. We interacted. David was in the lab at the time. Heidi Tissenbaum, who went on to do some really important work, was in the lab at the time. Brad Johnson, who went on to do really important work, was in the lab at the time. So there are all these really creative and smart people in this environment that was, you know, a little bit competitive, very supportive as well. And it was that mix, I think, that really had an impact on me. And it was also a time in the field when, you know, the field was growing rapidly. New tools had come online that for the first time really allowed people to do true mechanistic molecular work and identify some of the foundational mechanisms around the biology of aging. So I think it was a combination of all of those things that you know, that allowed several of us to really become immersed in the field and go on and have successful careers. And I hope my careers, I would like to think that maybe my biggest successes aren't behind me, but I think it was a combination of all of those things that allowed several of us to make a mark. But I think we also, because of that environment and because of where the field was at the time, we got hooked and stayed in the field. I think oftentimes you'll see people, you know, as graduate students who go on and do something completely different when they get to their postdoctoral research and their independent careers. Several of us who were in Lenny's lab around that time stayed in the field and didn't go work on something else. And so that's probably part of it as well.
Yeah, and it's so great that many of you have your own labs now, and you're creating a future generation of experts in this field that will hopefully, you know, go on and do even greater things.
Yeah, I think they definitely will. I say this whenever I'm talking to junior people in the field right now. I think this is such an exciting time to be a graduate student, postdoc, junior faculty in the field of aging biology or geroscience or whatever you want to call it, because there are so much renewed interest and resources coming into the field right now that I think the opportunities are just huge. I truly wish I was 25, 30 years younger and coming into the field now, because I think the opportunities to make, just amazing discoveries that are of high impact, they're almost unlimited right now. And so it's a really, really exciting time to be a young person coming into the field and doing science around the biology of aging.
Yeah, it was a great time when you were starting out, but arguably it might even be a better time now.
Yeah,
in this
absolutely.
case. One of the things in doing this summit, it's interesting talking to experts on longevity coming from all different places. One question I like to ask them is, what is your view of longevity, aging? Why do we age? What are the mechanisms? Is it wear and tear? Is it hyperfunction? All these things. And it's very interesting because there's no clear consensus from the experts. Everybody has a slightly different take on it.
Well, and I think that there's a lot actually to unpack there. So if the first thing I would say is when I think it's important to recognize that the word aging means different things to different people. And so oftentimes I think the disagreements that come up around, is aging a disease? What is aging? Really, you can track that back to how the individual person is defining aging, right? So I try to be precise in my language and talk about biological aging, because that's the way I think about it. There's chronological aging, which is just the passage of time. That's pretty easy definitionally to agree on. But biological aging is something different. And I think of biological aging as the physiological processes, molecular, biochemical processes that result in at least a permissive physiology as we get older for all of the different functional declines and diseases that go along with old age. And again, I've tried to be precise. I'm not saying necessarily that biological aging causes cancer, Alzheimer's disease, heart disease. I think it does to some extent mechanistically, but you can argue about that because we really haven't proven that. I don't think you can argue about whether or not biological aging creates a permissive physiology for all of those functional declines and diseases. And given that, I would say that biological aging is really where we should be focusing our attention if our goal is to keep people healthy as long as possible. So that's kind of the way I think about aging. Now, what are the mechanisms of aging? That I think the reason why there's so much disagreement or difference of opinion is we really are still figuring that out, right? So I think it's important to appreciate in the last 20, 30 years, scientifically, we've learned a lot about the mechanisms of aging. Again, molecular, biochemical, genetic mechanisms. But we've also really only scratched the surface. So we've gotten to the point where we can put some names to these things. I'm sure many of your listeners are familiar with the hallmarks of aging. And really the hallmarks of aging are just sort of a set of 9 or 10, depending on who you talk to, conserved processes that seem to be related to the biology of aging in every animal where we've looked. And so we can put names on these things. These are things like DNA damage, telomere shortening, epigenetic changes, senescent cells. Those are aspects of biological aging, but there's a lot that we don't understand underneath the hallmarks. And I would argue also outside of the hallmarks of aging, still yet to be figured out. And I think that's why you get differences of opinion or disagreements over what is aging, what causes aging, and that leads to some of the different theories that you alluded to, like hyperfunction, rate of living theory, right? Again, I think we don't know enough to really be able to say with a high degree of confidence, why did aging evolve? How did aging evolve? And what are all of the underlying mechanisms that are associated with biological aging? There's a lot to be learned. The last thing I will say on that, though, is we don't have to understand everything about biological aging to be able to have an impact on it. I think we know enough today that we can start to develop and test interventions that modify the biology of aging in a way that we predict will keep human beings and potentially companion animals healthier longer by targeting the biology of aging, slowing it, maybe even partially reversing some of the aspects of aging. So I don't think we have to understand the whole thing in order to be able to have an impact on it.
Yeah, you've made the point in several of your presentations, or you've underscored the point that aging is the single greatest risk factor for most of the chronic diseases that we face.
Yeah.
And small improvements in aging and longevity will dramatically multiply over all the chronic diseases.
Yep.
And so by that line of reasoning, then, if we come up with interventions to improve longevity and aging, we should expect to see those interventions applied not only to the phenotypes of aging and improvement, but also by the chronic diseases that determine longevity. So, and we'll talk about that in a moment as we get into one of the drugs here.
Yeah, but I would say we already know that's. I mean, the funny thing to me is people will sometimes push back against that. Well, you don't really know that. I'm like, yes, we do. We absolutely know about lifestyle changes that will reduce your risk for all of the chronic diseases of aging and improve your function as you get older. So there's really shouldn't be any debate about whether the biology of aging exists and whether it's modifiable. We already know you can do that. You can do that through exercise. You can do that through nutrition, right? So I think it's there's a little bit of a disconnect, and some people just want to argue, right? But I don't think we need to argue about this. This is a solved question whether or not there is a biology of aging. I think the other way to think about it is you can also just look across the animal kingdom. If you're at all skeptical that there is an underlying biology that regulates the rate at which animals age, just look at dogs, right? Everybody is familiar with the idea that, one human year is about 7 dog years. All that means is dogs age about 7 times biologically faster than people do. They get all of the same diseases of aging that we do. They're all age-related in dogs. They get all of the same functional declines that we do. It just happens about 7 to 10 times faster in dogs than it does in people. And that 7 to 10 depends on body size. So there's clearly this biology of aging, and it is determined by genetics and environment. And if we can understand what those genetic and environmental factors are, we can modify them in ways that will improve aging trajectories, keep people healthier longer, probably alive longer as well.
So let's talk about rapamycin. What is rapamycin, and why is it important for our understanding of aging and longevity?
Yeah. So rapamycin it's a small molecule drug that was first discovered on Easter Island, also called Rapa Nui. That's where the drug gets its name from, and it's produced by a bacterium found in the soil there. And we know the biochemical mechanism of rapamycin. It's a small molecule inhibitor of a protein called mTOR. mTOR, mTOR, actually stands for mechanistic target of rapamycin. So they named the protein after the drug. So rapamycin inhibits mTOR. So I think really the question to answer is, what is mTOR and why is it important? So mTOR plays this fundamental role in every eukaryotic cell that we know of. So again, for people who maybe haven't taken biology for many, many years, right, there are bacteria, which are typically prokaryotic cells, and then there are eukaryotic cells, which are all the cells in our bodies and in all animals. So every eukaryotic cell that we know of has mTOR, and mTOR plays this fundamentally important role, or one of its roles is in sensing the environment and then helping the cell or the organism make a decision about whether the environment is suitable for growth and reproduction. And one of the main things that mTOR senses is nutrient availability in the environment. And so from an evolutionary perspective, this is really important. Every organism that has ever existed has had to do this, has had to sense the environment, sense how much food is available, and then make a decision, is it a good time to grow and reproduce or not? And when there's lots of food around, from an evolutionary perspective, that's usually the successful strategy, is to grow as fast as possible, develop, reproduce, because you want to have babies when you have food to give them. The flip side of that is when there's not much food around, it's a bad decision to have babies. And so what mTOR does is it senses the amount of food in the environment, and then when there's lots of food around, mTOR gets turned on, and that tells the cell in the organism, okay, now's a good time to grow and reproduce. When there's not much food around, mTOR gets turned down, and that tells the cell in the organism, okay, we need to shut down growth and become stress resistant. And the mechanisms are pretty well known for how this works, but fundamentally, that's kind of the important gatekeeper role that TOR plays. So rapamycin is this drug that that tells TOR to get turned down, to sort of ignore the nutrient sensing cues and get turned down regardless of how much food is around. And what we and many other people in the field sort of found out many, many years ago, this was back in the early 2000s when several of us sort of independently, and in our case, accidentally, discovered that when we turn down TOR, we increase lifespan. And at that point, Brian Kennedy and I were working in yeast. Pankaj Kapahi was working in flies. A couple of other labs were working in C. elegans. And all of us independently found out that mTOR was this really potent regulator of lifespan in all of these different cellular and animal models. And then around 2009, a study from the interventions testing program showed that turning down mTOR in mice, and in that case, they used rapamycin, was enough to increase lifespan in mice. So this picture emerged that across a very wide range of different organisms, from single-celled budding yeast all the way up to mammals like mice, you could turn down mTOR, and that was enough to increase lifespan. And in mice, what we've learned since then is you can do this starting in middle age, which is really interesting, and we can talk more about that. And that you're not only increasing lifespan, but in pretty much every tissue and organ where scientists have looked, you can improve function during aging. So it's not only that they're living longer, but you're actually maintaining function of tissues and organs much later in life. And what's really exciting to me is that in at least some cases, you can reverse functional declines that have already happened with old age. So in my lab, we've shown this in the oral cavity. We can reverse periodontal disease with rapamycin in mice. Others have shown that you can reverse immune dysfunction, age-associated immune dysfunction in mice. Others have shown you can reverse ovarian dysfunction. Others have shown you can reverse cognitive decline. So, and I keep saying others, this is the other great thing about rapamycin and mTOR. It's not one lab. It's not only one lab doing this work. There are dozens of different labs around the world who all get the same answer. And so that provides a lot of confidence that this is a real and robust effect that is highly reproducible. And, you know, having been in this field for a long time, I'll just say, that's often not the case with a lot of what gets popular attention. It turns out to not be highly reproducible. Everybody agrees mTOR and rapamycin is a robust and reproducible target to modify the aging process in a positive way.
Now, mTOR, let me get this straight, is arguably the single most important signaling molecule protein in the animal kingdom, conserved biologically over billions of years, and it wasn't discovered till 1990 in the 90s. And
so I'm hesitant to state it quite that way. So here's the way I think about it. So look, personally, I agree with that, but I don't say it that way. But I actually think the way I would frame it is, we know that that mTOR is a key node in this network of interacting proteins, right? And so other things in that nutrient and growth promoting network include things like insulin-like growth factor 1, growth hormone, insulin, FOXO-transcription factors. There's a bunch of important factors in this pathway. It just turns out that some of them seem to be more amenable to modification in ways that have a positive impact on health span and lifespan. And mTOR seems to be a particularly good node in this network to tweak to positively impact health span and lifespan, at least in laboratory animals. Now you're asking, why wasn't it discovered, or at least in the context of aging, wasn't discovered till the early 2000s? And even before that was sort of understudied, you know, in the sort of broader biological world. I mean, sometimes that's just the way these things evolve, right? So part of the reason why mTOR became highly studied and we learned a lot about mTOR was because of this accidental discovery of rapamycin, right? So again, and people, I think a lot of times the general public doesn't appreciate how much of science is serendipity, right? It's not so much that we are really smart a lot of the times and go find the answer. It's that, you know, we're doing experiments and something unexpected happens, and it's those unexpected things that then lead to the big discoveries. And so this molecule rapamycin was discovered. Nobody knew how it worked, but what was observed was you put it on cells and they stopped growing. And so people thought, oh, that might be a really useful anti-cancer drug or a really useful antifungal. I wonder how it works. And so people started doing biochemistry and genetics to figure out when we put rapamycin on these cells, why do they stop growing? And that's how people identified TOR. And then once TOR was identified, then many labs started studying it, and we learned a lot about how it interacts with all the other players in that network. And the same thing was true with discovering that TOR and rapamycin were important for aging biology. At least in our case, we were doing what's called an unbiased genetic screen for factors that influence lifespan. So it's not like Brian and I went into this thinking, oh, TOR must be really important for aging. It was because we had a tool that allowed us to look one by one at individual genes in the genome and ask which genes affect lifespan. And we got lucky that in the first 500 that we looked at, TOR was there. If it wasn't, it would have taken us longer to figure it out. But TOR happened to be in that first set of 500, and we saw that when we turned down TOR, all of a sudden lifespan was extended. And then, I was like, well, what's TOR? I didn't know anything about TOR. And it turned out there was this drug, rapamycin, that inhibits TOR. And well, maybe rapamycin would have the same effect. So, it's really this serendipitous sort of process that led us to discover that rapamycin could increase lifespan. And I think that's probably true for these other labs that were looking at mTOR and rapamycin as well around the same time.
Yeah, you mentioned that caloric restriction is reliably the most reliable way to improve longevity over the years and in studies. And now intermittent fasting is getting a lot of publicity on that. How does rapamycin work, suppressing mTOR as nutrient sensing compared to caloric restriction or intermittent fasting? Are we looking at the same mechanism there?
Yeah. Really good question. I would say again, there's a little bit of debate about to what extent does rapamycin mimic caloric restriction. So here's the way I think about it. They are overlapping but distinct interventions. So we know that one of the most potent things that caloric restriction does is turns down mTOR. And this gets back to what I was talking about before. mTOR is a nutrient sensor. So when people or animals fast or are chronically calorically restricted, mTOR activity goes down. And we know rapamycin turns down mTOR. The difference, I think, arises from the fact that caloric restriction does about 10 million other things in addition to turning down mTOR, while rapamycin is a very specific mTOR inhibitor. Now, there are lots of effects of rapamycin that are downstream of mTOR because mTOR regulates a bunch of stuff, but caloric restriction in some ways is what I would call a much dirtier drug. It has many other effects that rapamycin doesn't have. And I think it's still an open question, to what extent are the effects of caloric restriction on lifespan and health span in laboratory animals due to inhibition of mTOR versus non-mTOR effects. And then there's this interaction between these other things that caloric restriction does that happens in the context of mTOR being turned down. And that still hasn't been figured out. I think there are a couple things to say about caloric restriction and fasting, though, that most people don't appreciate. So caloric restriction is definitely the most robust, and by robust, I mean the largest effect size intervention for increasing lifespan. And you could argue health span a little bit, but I'm just going to make a blanket statement that's not completely precise that caloric restriction improves health span in laboratory animals as well. It has the biggest effect. I don't think anybody would argue that for non-genetic interventions. You can get up to about a 60% increase in lifespan in mice if you restrict calories by 60%. What's often not talked about or appreciated is in about 1/3 of genetic backgrounds where caloric restriction has been tested, it has no effect on lifespan or actually shortens lifespan. So there's a genetic component to whether or not, and again, this is in mice, whether or not caloric restriction increases lifespan. The fasting sort of myth that people don't understand is in laboratory animals, at least, fasting only increases lifespan if the animals are also calorically restricted. If you do intermittent fasting in an isocaloric setting, the impact on lifespan is somewhere between 0 and 4%. So it's tiny. So there's this idea out there popularized by certain people that intermittent fasting is universally going to be good for health in humans. I think intermittent fasting is a very useful strategy for some people to lose weight or maintain weight. It's unclear whether or not intermittent fasting in the absence of caloric restriction does anything in laboratory animals or will do anything positive in humans. That's my personal view of the literature, but it just unfortunately gets communicated in a very misleading way, I think, often for the general public. this idea that intermittent fasting is this really potent way to target the biology of aging, that's only true in laboratory animals if you also restrict calories by between 30 and 50%.
So given that, as you mentioned, in laboratory animal studies that rapamycin not only in some cases slows the hallmarks of aging or phenotypes of aging, but in some cases actually reverses it. And given the fact that it can be delivered, at least in the ITP model, at middle-aged mice rather than lifetime exposure, and this is in animals now, what is the pushback to people using that for humans? Obviously, more work needs to be done, but why aren't more people aware of the potential of this drug?
Yeah. So I think, I would say there are really two things that are a barrier to sort of more wide understanding and maybe use of rapamycin. I mean, I think we need to be honest, right? We still don't know in humans whether or not rapamycin is going to have the same effects on lifespan and health span that it does in mice. I think we could probably spend an hour talking about the data around that, but we don't know for sure, right? There haven't been double-blind, placebo-controlled clinical trials, or at least very many of them, to try to answer those questions. So there are really two things I think that have been barriers. One of them is that rapamycin in the clinical world, it goes by the name sirolimus or Rapamune, has sort of a bad reputation because of the way that it was developed and approved by the FDA. So rapamycin, again, sirolimus, is an FDA-approved drug. It's been approved, I think, since 1997, something like that, 20 plus years. And it was approved as an organ transplant drug. So at high doses, daily doses, rapamycin will help patients who have had an organ transplant in combination with immunosuppressants, not reject that transplanted organ. So that's how it's been used. And in that context, there are a long list of side effects that go along with taking rapamycin. So it has sort of a bad reputation among clinicians, and it has a bad reputation at FDA as a drug that has lots of side effects. So I think that's been one of the real barriers to people even psychologically thinking about rapamycin as a drug that healthy people or healthier people who are aging normally, maybe, let's say not organ transplant patients, might take to modify their biology of aging in a positive way. There's a lot of pushback against that because the gut reaction is that's a dangerous drug, right? So I think that's one thing. The other thing is that it's really, in the timeline of scientific discovery, fairly recent that we've learned about these effects of rapamycin on health span and lifespan, even in laboratory animals like mice, right? The first study for lifespan was 2009. I think the first study showing that you could increase lifespan with a short-term transient treatment with rapamycin came from my lab, and that was in like 2000. I should remember this, 2016 maybe, when that was published. So in the timeline of science, this is all happening really, really recently. So it's only started to become appreciated, how widespread the effects are in different tissues and organs, and that you don't necessarily have to start taking the drug as a teenager if you were a person, and we extrapolate from mice to get some of these effects. So we're learning a lot in the last few years, and there are probably now, somewhere between 1,000 and 10,000 people around the world, I would guess, who are taking rapamycin off-label for potential effects on health span and lifespan. And I know you've been involved in trying to collect information from some of these people. I've been involved in that as well, to start to try to learn, anecdotally, it's going to be imperfect data, but start to try to learn what are the real side effects look like, what do maybe the real benefits look like for people who are taking rapamycin, with the idea that it might have a positive impact on their health span.
Several other of our interviewees have mentioned biological clocks, sort of DNA epigenetic methylation clocks. It's always hard to get longevity data on human beings. What have these been used with rapamycin? What's your take on that? Are they valuable?
Yeah, so I would say, I think it depends on so this word clock is sort of a catch-all for biomarkers of longevity or biomarkers of health span. That's really what we're talking about. Can we use different phenotypes to assess whether or not somebody is aging at a different rate? That's the ultimate goal. And a variety of different types of data have been proposed as useful biomarkers or clocks. So epigenetics is one. There's blood biochemistry you can do that correlates with future health outcomes is another. I really like functional measures of aging. I think that's what's really important. So I would put those in the category as well. How well are you functioning? And there's a variety of different organ functional measures that you can do. There's physical functional measures. So I think the answer to your question, though, is conceptually, yeah, these things can be really useful. What we don't know yet is what are the best set of biomarkers to put into, what I think we all would really like, which is an integrative clock that is telling us overall how well is somebody aging at the moment, and maybe more importantly, how well are they going to age in the future? So it's pretty early days with these clocks. I'm not convinced yet that we have biological aging clocks that are actionable. And by that, what I mean is, what I would really like is a test that I could take on myself or give to someone else and say, if you take rapamycin and it moves the needle on this test up or down, that's a good sign. And you should keep taking rapamycin. If it doesn't do that, that's a bad sign and you should stop taking rapamycin. That's what we really want, right? We want actionable tests that will allow us to make personalized recommendations for people to get them on the, something closer to their optimal aging trajectory. I don't think we have that yet. And I think, in some ways, the marketing has gotten ahead of the science, where there's a bunch of these direct to consumer biological aging tests that people can go buy. I don't have a lot of faith in any of them right now that they're actually telling you information that's useful. I think we'll get there, but I think I think we're still a little bit early to know which of these tests are going to be most informative. That's something I'm working on right now, and I'll probably be working on for the next couple of years, is trying out some of these different biological aging tests and trying to get closer to some sort of, integrated signal that I feel confident about.
It seems like one function of mTOR as a sort of a master survival switch is to turn from growth when nutrients are present, and then when nutrients are not sensed, turn to sort of protection mode.
Yeah.
Turn down inflammation, autophagy goes up, all those things. Thinking about the mind and stress, one model for stress is that chronic stress, I guess acute stress can be good for longevity, but chronic stress shifts our bodies from growth to protection. And that seems like that's opposite of what we want to do with mTOR. Does that ring any bells for you, or it's a little bit off topic?
Yeah.
But
I think you're right. Again, conceptually, from a general sense, that's all true. I think this word stress is a catch-all, right? So that can mean a lot of different things. So when we talk about rapamycin and turning down mTOR, inducing a stress resistant state, what we really mean is that, at least for certain types of acute stress, it makes cells, organs, tissues less likely to be damaged by the stress, right? So stress itself isn't a bad thing. It's the consequences of stress and the damage that it does to organs and tissues and cells that lead to the functional declines and diseases that we associate with chronic stress. And so I think that we have to appreciate that turning up protective mechanisms that make cells more resistant to stress is part of the chronic stress response, but it's not the only thing that's happening, right? So when you have, for example, you're constantly exposed to radiation, let's just say, you get exposed to radiation, your body, your cells are immediately going to turn on a protective response that helps keep those cells, to the best of the cell's ability, from being damaged by that radiation. But if you keep being exposed to more and more radiation, eventually you're going to overload those protective responses, and then you're going to accumulate damage. So the stress itself ultimately is what's going to drive the damage, but it's only when you overload the protective responses. Rapamycin, in some ways, turns up the protective responses, but you don't necessarily have the stress itself driving that response. So one way to think about it is you're preconditioning maybe the cell and the tissue to be more resistant if it gets hit by the damage before the damage gets there. That's sort of hand wavy, but I think that's part of what's going on. But again, I think there are a variety of different types of stress response mechanisms, and I certainly wouldn't want to suggest that rapamycin is turning up all of them, right? So I think we have a lot to learn about, which protective mechanisms are being affected by mTOR and which are mTOR independent, and how does that integrate with longevity and aging. Again, this goes back to what I was saying before. There's a lot we don't understand, a lot to still be figured out. And even this idea of inflammation, I think, is complicated, right? So, we know that with aging, there is an increased amount of what we call sterile inflammation, which I think the easiest way to think about that is your immune system reacting to stuff that it shouldn't be reacting to. So we need our immune system, we need inflammation to respond to infections and things like that. But with age, we get this dramatic increase in sterile inflammation, which is your immune system responding to things that it shouldn't be responding to. And rapamycin is quite potent at knocking that down. So that's a good aspect of what rapamycin does. If you have too much suppression of inflammation, though, that's where you get into the realm of immunosuppression, which is what rapamycin is used as in organ transplant patients. So there is this balance and this dose response with stress resistance and anti-inflammatory effects where, there's a sweet spot where we would want to be that gives you the benefit without so much of the detriment. But we don't know exactly where that is. And if you push it too far, you may get into the point where you're actually having negative effects in addition to the positive effects. I think that's true with inflammation and immune function. I think that's also more generally true with stress resistance.
Is there any evidence for interactions between psychological stress and mTOR or rapamycin? Any interactions there in the psychology space?
So I don't know the specific answer to the question about psychological stress. So I'll say a couple things that I do know. One is that there's absolutely clear mechanistic connections between chronic psychological stress and the biology of aging. I think there have been several studies that have looked at chronic psychological stress and telomere shortening. Telomere shortening is one of the hallmarks of aging. Clear evidence that people who are under high levels of psychological stress tend to have shorter telomeres. You can find many other connections where it seems clear that psychological stress can accelerate to some extent the hallmarks of aging, which is consistent with the idea that they're biologically aging more quickly. And when you think about it, that's not shocking, right? Again, we know that people who are exposed or experience high levels of chronic stress throughout their lives tend to have a variety of poor health outcomes later in life, consistent with the idea that they're aging more rapidly. So there are absolutely those connections. And then there are these connections between rapamycin and brain function that I honestly don't understand, but have fascinated me. So, and let me put a little bit more meat on that. So we know that changes in brain function with aging, right, happen. There are declines in cognitive function, changes in cognitive function that happen during aging. In some people, that manifests itself as dementia or things like Alzheimer's disease, which is a subtype of dementia. In animal studies, rapamycin is very potent at reversing or preventing some of those age-related changes in brain function. That makes sense in the context of aging biology. But then there's this whole other body of literature on rapamycin that's kind of evolved in parallel to the studies in the biology of aging, where rapamycin has potent effects on autism, for example, or on seizure-related diseases. And I don't understand that. So let me say this. Let me be careful how I say this. I speculate that there are mechanistic similarities to what rapamycin is doing in the context of dementia and normative brain aging, and why rapamycin has these effects in autism and seizure disorders, but I don't know what those mechanisms are. But I speculate that they're important and probably tell us something really fundamental about how mTOR is acting in the brain that we don't currently understand. And then there's this emerging sort of literature that I'm peripherally aware of on rapamycin in combination with some psychedelic drugs or things like ketamine, where you get these really interesting interactions. For example, there's some evidence that ketamine, that rapamycin potentiates the effects of ketamine on things like depression or pain. And again, I think that's going to be related to these observations that rapamycin has effects on autism spectrum disorders and seizure disorders, but I don't know what the mechanisms are yet. So I think there's a bunch to be learned there that we don't really understand. Right now, we're kind of at the observation stage where we see that there are these effects and interactions, but we don't mechanistically really understand how they're working. At least I don't.
Yeah, it's so many unanswered questions. I'm reminded of Chris Palmer's work from Boston. He's a psychiatrist who's taking some of his schizoaffective disorder patients and on serious medications, and he's putting them on a ketogenic diet, putting them into ketosis. Not all of them, but some of them will respond to the point that their medicines are already dropped. And if you you think turning mTOR down, just remove the glucose from the diet and switch to ketosis, you'll effectively turn mTOR down. Yep. Maybe that's a common mechanism, but who knows? Like you say, we don't even, we're just scratching the surface.
I think there is something there, though. So I'm glad you brought up the ketogenic diet, because that also is quite potent in seizure-related disorders, at least some patients, right? So again, I think you're right. I think that mTOR is a common denominator there that probably is important. But it's, again, ketogenic diet's kind of like caloric restriction in the sense that it's going to hit mTOR, but it's going to do a bunch of other stuff as well. And so you have to be careful not to say that all of the effects of the ketogenic diet are through mTOR. Some of them probably are. Yeah, I think the interactions with ketamine in particular are a fascinating area that it'll be interesting to see how this evolves. So, I have a colleague here in Seattle who's a psychiatrist who has been treating some of his patients with ketamine plus rapamycin, and he's told me some really profound stories about improvements in his patients. Particularly things like chronic pain, and it's reminiscent of what you were saying with the ketogenic diet, where sometimes people come off of medications that they've been taking for decades, right? When they go on this combination, they don't have to take the chronic pain medication anymore. They have the strong antidepressants. So, yeah, there's clear interactions there that are going to turn out to be really important, and it's going to be fascinating to see how that evolves, and also whether or not, to what extent are those related to the way that rapamycin is impacting the biology of aging.
Yeah, there's so many projects to talk about, but one project that you're working on, I wanted to mention because our audience can actually participate in it, or at least their companions can. Could you talk about the dog aging project? It's so interesting.
Sure. So the dog aging project is a large scale, what we call longitudinal study of aging in pet dogs, companion dogs. So what we are doing is really trying to understand the biology of aging in companion dogs with the goal of being able to help our pets live longer, healthier lives. And so I can say, I think generally maybe the way to think about it, there are really two pieces to the dog aging project. This is an oversimplification, but it's an easy way to think about it. There's an observational study, which is very, very large. We have about 40,000 dogs in the observational study right now. Any dog is eligible for the observational study, any age, any breed, any size. And the goal there is to understand what are the most important genetic and environmental factors that influence healthy aging in our pets. So if any of you have dogs, I'd encourage you to go to dogagingproject.org. There's a little button, nominate my dog, up in the upper right-hand corner of the website. Join the Dog Aging Project pack, and you will be part of what is one of the largest, if not the largest, community science, open science projects in the world, with the goal of helping our pets live longer. The other piece of the dog aging project is to do something about the biology of aging. So we have a smaller, double-blind, placebo-controlled clinical trial of rapamycin in dogs to really try to answer the question, does rapamycin increase health span and lifespan in pet dogs? So to be eligible for the rapamycin trial, dogs have to be at least 7 years old, between 40 and 120 pounds, and can't have any significant pre-existing health conditions, because it's a study of healthy aging. And we are recruiting for both the longitudinal study and the clinical trial. So again, if anybody's interested, please consider going and nominating your dog for the study. I'm obviously particularly excited and interested to find out, to what extent does rapamycin have an impact on healthy aging in our pets?
Yeah, and I know you're already mining that data, the dog trial data. I know one interesting observation you make was that dogs who were fed once a day versus dogs that were fed twice or 3 times a day had a different rate of chronic disease complications. Or maybe I'm not
Right.
summarizing that correctly.
So
What does
that's
that
pretty.
say for humans?
Yeah,
Yeah, go
yeah.
ahead.
I mean, so first of all, let me just be a little bit precise. So
Yeah,
you were
please.
close. But again, I think it's really important to appreciate, because this is an observational study, we're very careful about correlation versus causation.
Right.
That study that you're alluding to, though, the way that analysis was done, these are owner-reported pre-existing diagnoses of disease or conditions. And the question we asked was very simple. We thought dogs might be an interesting natural model of time-restricted feeding, because some owners feed their dog once a day, some twice a day, some 3 times a day. Some actually feed their dog what's called ad libitum. The dog has free access to food. So the simple question was, are dogs where the owners only fed them once a day, are there any differences in the likelihood that those dogs were diagnosed with one of 10 different age-related categories of disorders? That included cognitive function, kidney disease, you can go down the list. And I didn't think this was going to work, because I'm not a big believer in time-restricted feeding, but as it turns out, I was wrong. So what we found was that in all 10 cases, the arrow direction of the effect was going towards dogs that were fed once a day were less likely to have been diagnosed with any of these 10 conditions. And I think in 6 or 7 of them, it was statistically significant. In a couple of cases, the effects were pretty big. So what that tells us is that, this is now looking backwards in times. Had the dog been previously diagnosed with these disorders, if the dog was fed once a day, the likelihood of a previous diagnosis was lower. Does that mean that being fed once a day caused them to not be diagnosed? We can't answer that. And here's where I think you do have to be careful, because there are obvious potential explanations for this correlation. One being that dogs fed once a day compared to 2 or 3 times a day might be less likely to be obese. And we know that obesity is a predisposition for multiple age-related comorbidities. So we have to do more work to figure that out. But it is intriguing, and it certainly may suggest that feeding your dog once a day means that it's less likely your dog's going to get any of these 10 diseases or one of these 10 diseases. It may also suggest that this is a useful strategy in humans, or at least in some people. But I think we have to be really careful not to jump the gun. It's a hypothesis generating observation that requires additional study to really get to definitive. I know that frustrates people. It frustrates me, but that's the reality of the situation. We can't say that there's causality there.
So many questions, but not enough answers. But it is an exciting time. Matt, maybe you could tell people how they can reach you on social media, how they can sign up for the Dog Aging Project if they'd like, and how they can find your website.
Sure. So my website is kaeberleinlab.org, and I'm on Twitter at @mkaeberlein, so first initial, last name. And then the Dog Aging Project website is dogagingproject.org. I'm pretty easy to find online, so I'm sure if anybody really wants to find me, you can find me online. But
Right.
I certainly, again, will make the plug that if you have a dog and you love your dog and you believe in science, please consider participating in the Dog Aging Project.
Yeah, very, very worthwhile. And a very creative approach to adding knowledge to this space. So thanks again, Matt, for spending an hour with us today and sharing your knowledge. And thanks also for all the great work that you do in your lab. And we're expecting great things from you in the future.
Okay, thank you. Thanks for having me. This is a great service that you're doing.
If you are enjoying this program, please hit that subscribe button, or even better, leave a review. Your support makes it possible for us to create the quality programming that we're continually striving for.
Can I start? Is it recording?
It's already recording.
Oh, sorry. This is for general information and educational purposes only, and it's not intended to constitute or substitute for medical advice or counseling. The practice of medicine or the provision of healthcare, diagnosis, or treatment, or the creation of a physician-patient or clinical relationship. The use of this information is at their own user's risk. If you find this to be on the value, please hit that like button to subscribe to support the work that we do on this channel. And we take your suggestions and advice very seriously, so please let us know what you'd like to see on this channel. Thanks for watching, and we hope to see you next time. You should say that.
That's good. It's for you to say that. That's very good. You like it? You want to do it one more time, or is that good? I think that was good.
Yes. You need to save the recording.
Very good.